I think we can go ahead and get started. I'd like to welcome everyone to our HIRN webinar. Today's title is A Roadmap to Restoring Tolerance in Type 1 Diabetes, CAR Tregs, and beyond. My name is John Qizhi, and for those of you that don't know about HIRN, briefly, this was and is a network that began in 2014. NIH started this network to address various aspects of type 1 diabetes. And what they did was they created different themed groups and organized these themed groups together as a network. And you see the names of these consortium, and each one is working on something a little different. All in all, the network is still growing. We have over 200 investigators, nearing 100 different institutions, and these are labs from all around the world. Now, we do have a website where we're constantly updating information as we get it, but today I just want to briefly draw your attention to a few things. First, of course, there's funding, and periodically we will release funding announcements. NIH will have announcements, other colleagues may have announcements, and there may be even opportunities in HIRN. So you can always check that funding tab to do that. And then we've got other ways that you can stay updated with the happenings in HIRN, and these are some of the tools. And you're here, obviously, you know about the these meetings. We have a lot of resources that are available to you, most of them in HIRN, some of them in other networks, and so we're highlighting that for you today. And of course, you can follow us on all of the social media channels that are available to you. One of the things that we have, and you may be curious about, is what we've called the HIRN resource browser. And essentially what this is this is a listing of the different tools and reagents and data sets and protocols, things that are either being used or generated by HIRN. You can find those here and access those here. Also, as I said, today's webinar and ones that we've done in the past, they're all recorded. They're all on YouTube. You can access them directly through our website, and of course, you can enjoy them here as well. Some of the opportunities I was telling you about, there's a couple of awards that have been released. You may or may not know about these opportunities. You can apply these. These are directly from NIDDK. So if you want to subscribe to our newsletter, that again, you can get that, and we let you know about these various things that are happening. So with that, we'll touch on these topics today: what role Tregs play in type 1 diabetes, and the history of Tregs, and where's the field headed. I think everyone should already be muted, and so please keep it that way while Dr. Ferreira gives his presentation. We'll have plenty of time after the presentation to ask questions, and I would actually encourage you to ask your questions live to him. If you don't want to do that, and if you just want to type it in, we'll read those as well. So with that, I'm gonna switch off and hand to Dr. Ferreira, and as we do that, I'm going to just give a brief introduction to him. Dr. Ferreira is an assistant professor in microbiology and immunology at the Medical University of South Carolina. He got his his bachelor's degree, I should say, in biochemistry at the University of Coimbra in Portugal. He then went on to earn his PhD in the same from Harvard University, and then did his postdoc work at UCSF. Dr. Ferreira is a T-cell biologist, but if you ever talk to him, he's a library of knowledge in science, and some of you may know exactly what I mean. But he's dedicated the last few years to studying CAR Tregs in type 1 diabetes, and I think that's what he's going to spend the good part of today telling us about. Dr. Ferreira. You are muted, Dr. Ferreira.
Yeah, there I am. Sorry. Hi,
There you
everyone.
go.
Thank you for the very kind introduction and also for the invitation, the opportunity. It will really be a treat to take everyone on a journey on how we may be able to use Tregs to treat type 1 diabetes. So I'll minimize my image here and off we go. So first things first. So the problem at hand is a very complex one, is autoimmune disease. And if you think of the immune system as an army that protects us from infections and cancer, autoimmune disease is kind of the immune system going rogue. Is a body at war, but this time with itself. And so there's many, over 80 different autoimmune disorders, as psoriasis, rheumatoid arthritis, vitiligo, thyroiditis, multiple sclerosis, and of course, also type 1 diabetes. And all these disorders have to do with unwanted destruction of tissues by the immune system. In type 1 diabetes, the target is the pancreas. The pancreas is this banana-shaped organ that sits in the middle of your body and has many functions. It has an exocrine function of secreting enzymes that help with digestion and an endocrine function that have to do with glucose regulation, with blood sugar controlling. And so in type 1 diabetes, the target is not really the whole pancreas. It's just this islets, these rosette-like structures, the Langerhans islets. And these islets have got alpha cells, beta cells, and others. And the target specifically is the beta cell, which are the cells that produce insulin that controls blood sugar. So type 1 diabetes patients then, what happens is that their islets, they're attacked by immune cells, by CD4 T cells, CD8 T cells, and others. And so what happens is, after the disease first starts, you then seeing that over time, the beta cell mass keeps going down and down and down. And you see in this histology slide here, pretty, dramatic representation of this reality, which is you start off with your islet in the pancreas, a lot of alpha cells that make glucagon and beta cells, so alpha cells in blue and beta cells in red. And then you see over time, the beta cells specifically disappear, and all there is left is the blue cells, is the alpha cells. And this destruction is carried out by immune cells, which are labeled in brown with CD45, for those who want to know. And so those are the cells there. And so we've known for a very long time, there's a very old chart down here. It's called an Eisenbarth graph, showing that, again, over time, we know that there's to be some precipitating event, some trigger that causes type 1 diabetes, which then makes the beta cell mass go down and down over time to a point where these patients rely on constantly measuring their blood glucose and injecting insulin because they don't make their own. And so now it's become more of a chronic disease because you have synthetic insulin, but still, if you don't inject enough or enough times, there's many risks. There's neuropathy, even limb amputation, going blind. And so the quality of life would be greatly improved if we could provide the curative treatment other than just daily management with insulin injections. So how could we do this? The way we could do this would be to actually address the root cause, which is the immune system, right? The problem started because this type 1 diabetes patient's immune system decided to recognize beta cells as foreign, as dangerous, as something to eliminate and destroy them. So how could we somehow re-educate, reset the immune system? Well, we'll have to understand how to work with immune tolerance. So tolerance is unresponsiveness towards a specific target, a specific antigen. And so this was first found a long time ago. This dates back to the 50s, but in the context of autoimmune disease and T cells, it was really in the 90s that a big breakthrough was made. And so it's this idea that, if you take mice and you take out the thymus very early on, the mice are just immune deficient. Their T cells can't go to school. They don't have trained T cells. But if you take their thymus 3 days after birth, now they actually get the opposite problem. Instead of being immunodeficient, they actually have autoimmunity, and they have gastritis, thyroiditis, ulceritis, and also autoimmune diabetes. And so there must be something happening very early on in the thymus that is responsible for the immune system as we know it, right? A sharp tool that attacks what is foreign and leaves alone what is self. And so this self-non-self recognition in this immune tolerance seems to be mediated by this group of T cells marked by the surface expression of CD25. So you see in these graphs here, if you are able to purify, to sort, FACS sort, CD25 positive cells, you then see a much lower incidence of autoimmune disease induction into mice that are transferred with these cells. You see this little schematic here from a review article in the 2000s where you see if you take a mouse and put just these CD25 negative non-regulatory cell fraction, the mice get autoimmune disease. But if you put everything together, the mice have no disease. And this is how we live, right? We have a mix, the perfect mix of T cells, of regulatory T cells and conventional T cells that prevents us from developing autoimmune disease. And so CD25 turns out is an alpha chain of the IL-2 receptor, so it's a high affinity receptor that controls IL-2. And so this will come in handy later down this presentation. So this is the first, proof in the mouse, but now we're humans, so we want to know, okay, do we also have these cells? And turns out we do. There were 6 back-to-back papers in 2001 showing that humans have the same exact CD4 positive, CD25 positive T cells in blood. And some people even went as far as they could expand them in vitro a little bit and show their suppressive function. So humans also have these regulatory T cells that are kind of the general of the immune system, right? They make sure nothing goes overboard, everything is under control. So now that we have these cells as well, okay, where do they come from? And are they really a cell type, or are they more of a cell state? Something that a T cell maybe goes through. And the answer is that is actually a defined cell type. And how do we know that? Because they have their own transcription factor, right? Transcription factors bind to DNA, turn on genes. And so they pretty much define cell types. And so now we know that Tregs, regulatory T cells, develop in the thymus, just like every other T cell. But what happens is, during thymic education, the reactivity of T cells has to be tuned, right? If a T cell has very low affinity to the antigen, binds very poorly, it gets thrown away. It's called death by neglect. No one cares about that T cell. It's useless. If it binds too strongly, though, that's also not great, because if it binds too strongly, remember, education has to be done on our own antigens, our own proteins. So if something binds too strongly to self, it could cause autoimmune disease. So then that cell is deleted. That's the negative selection there. Now, there's kind of a range where the affinity is higher than average, but it's not so high as is to be deleted, to be killed. And that's where the CD4 positive, FOXP3 positive Tregs come from. And so there's this distinct cell type that turns on this transcription factor called FOXP3, which is also found in adult Treg cells in humans and in mice and many other animals. And so this is what keeps the transcriptional program, this gene program that allows for this to be a separate cell type, whose function is not to kill virus-infected cells or cancer cells or stress cells, but to prevent T cells from going overboard and killing after their immune reaction has already come to an end. And so there is a specialized function, right? Our immune system as a cell, whose function is to suppress specific immune responses. And I'm really emphasizing this because I think it doesn't get enough attention in the general media. People usually want T cells to kill stuff, to help us get rid of stuff. But it's equally important to make sure that when the killing has stopped, it can come down to an end to avoid unwanted damage. And of course, in an extreme, to avoid autoimmune disease as a start. So we have this FOXP3 Tregs, and how do they work? Well, now we know quite a bit about their molecular mechanisms. One I just mentioned was C25, the IL-2 receptor alpha chain. And why I mentioned this, because this is one of the primary mechanisms by which Tregs work. Interleukin-2 is an important T cell growth factor. It's actually how it was first named, TCGF, T cell growth factor. And Tregs, what they do is they don't make their own IL-2, unlike conventional T cells. And on top of that, they always express high levels of these high affinity chains. So they soak up whatever the Treg is, it will soak up all the IL-2 so that conventional T cells don't stand a chance to never mount an immune response. We also have other mechanisms. One of them, for example, is CTLA4. So CTLA4 is a very interesting molecule because this is how the Treg binds directly to an antigen-presenting cell, which is kind of the cell that informs T cells to go find their target cell. And so what a Treg does with CTLA4 is that it binds very strongly to these co-stimulatory molecules, we call them CD80, CD86. And by removing them, this antigen-presenting cell is now a tolerogenic, tolerance-inducing APC. Instead of being an inflammatory cell, it becomes a tolerogenic cell. And so in that process, now the Treg is basically modulating the immune response to become phased out. Tregs also control the metabolic environment. If there's ATP flowing around from cells that just died, that's usually an inflammatory signal, right? Every time there's cell death is inflammation, you want to clear that up. What a Treg does, though, is a Treg will convert extracellular ATP into adenosine which is a potent immunosuppressor that binds to and inhibits T cells. So once again, the Treg is basically taking an inflammatory environment and converting it into a suppressive environment. And another mechanism that is perhaps less talked about is that Tregs themselves can also kill. They're not going to kill the virus-infected cells or anything, but they're going to kill antigen-presenting cells, right? So for example, a B cell or a dendritic cell. So, right, one way to stop an immune response is to stop the antigen-presenting cell that is feeding the immune response to start. And so that's another very important mechanism, we think, by which Tregs can really reset immune responses and stop the priming. They don't even let T cells get started. Okay, so with all these different molecules, right, how can we do this, So we know that Tregs are very important from human data. So, nature does the best experiments. And so there's this syndrome called IPEX, which is a monogenic form. So I'm emphasizing that, right? It's a monogenic form of autoimmune disease. Usually autoimmune disease has many genes, many environmental factors, but this one, a mutation in FOXP3, is enough to cause autoimmune disease. And that's because these patients have no functioning Tregs. And interestingly, what is the most common by far disease in IPEX? Type 1 diabetes. And so this is how we know, if you will, that Tregs is a potential avenue to treat type 1 diabetes, because if you don't have Tregs, the first thing you get is type 1 diabetes. So it stands to reason that if you are to replenish, to refurbish, to give more Tregs, the right kind, we'll go about that in a second, we could treat this disease, this type 1 diabetes. And then the other kind of clue, not as clear cut if you ask me, is that if you look for genes involved in polygenic type 1 diabetes, several of them are actually Treg genes. You see in this bar chart here, PTPN22 is downstream of IL-2 receptor signaling, C25 itself, as we just mentioned, and CTLA4. And if you go on this tiny table here, I made the important genes bigger font, you see again the same thing, C25, CTLA4, also IL-2 itself, and neuropilin-1 and RP1 is also expressed in Tregs. So several genes in polygenic T1D are Treg genes. And so together with the data from, IPEX, we really think that Tregs play a key role in development and hopefully therapy of type 1 diabetes. So then that makes many of us believe, well, if these Tregs act by so many molecular mechanisms that it would be hard to replicate with individual molecules, and they're so important for development of type 1 diabetes, then can we use them as living drugs to treat type 1 diabetes, to take situations where there's excessive immunity, in this case against the beta cell, to a case of there is a regulation, a scenario where there's no autoimmune attack. And so how far are we with this? So this is a pretty long timeline, but I like to show this slide because just to show you how fast the Treg world has advanced. It was only in the 90s, 1995, that paper came out that I showed you where, there's this new cell type, express C25, and prevents autoimmunity in mice. And then not that much later, all those different papers, 6 different papers, show that humans also have this cell type. That was 2001. Fast forward, 10 years, 2011, and they're already treating people with these cells. There's a first clinical trial for graft-versus-host disease in 2011. There's a trial for type 1 diabetes in 2012, and there's another one. And so this shows us how fast the field has moved. But how about type 1 diabetes? So type 1 diabetes therapy with Tregs was really first to be, potentially work in mice. And so in mice, we have this non-obese diabetic mouse, NOD. And so this NOD mouse kind of allows us to see the disease develop. So a lot of studies, NOD mouse, they ask the question, if we were to catch type 1 diabetes early enough, could we prevent it? And so sure enough, in this paper, this was back in 2004 by Qizhi, Tang, and Jeff Bluestone, they actually found a method to robustly expand Tregs. And it is a big deal, 20 years ago now, because people could not grow Tregs because they need so much IL-2 and other growth factors. So they're able to grow them in vitro, and they had to be antigen specific. So BDC 2.5 is an insulin-specific T cell receptor. So 100% of these Tregs are laser focused on recognizing insulin. And so what you see is very few cells, only 500,000 cells is enough to prevent autoimmune diabetes development in these NOD mouse. Moreover, if you are to inject more cells, 10 million cells, so 20 times more, you can actually revert type 1 diabetes. So if the mice already have type 1 diabetes, you get roughly half of the mice to be cured, at least for a very long time, for up to, this is up to 20 weeks of after injection of the 10 million Tregs. And so these really show that okay, we know that Tregs are important in the development of type 1 diabetes, and now we show in the mouse that if we have insulin-specific, so islet-specific Tregs in the mouse, it can actually prevent type 1 diabetes and actually revert it if you give more cells. So then the field is like, okay, can we use this then for type 1 diabetes in humans? So there's a plethora of trials, not as many as other fields, but still there were 50 back in 2019. I think now we're closer to 100. And there's these main three categories I mentioned: graft-versus-host disease, organ transplant, and autoimmune disease. And so we more focus on autoimmune disease, specifically type 1 diabetes. And so we focused on that. So how is it looking like in humans? So phase 1 trial, again, by the Bluestone group, it showed that you can infuse up to 2.6 billion polyclonal Tregs, and it's safe for type 1 diabetes, no adverse effects. Now, however, even though this was safe, efficacy was not really proved. Now, of course, it's a phase 1 trial, but you can look at C-peptide, which is a more stable proxy for insulin, and you see different patients. That some patients, it keeps going down as it was before, so they still cannot make insulin. Some is kind of looking flat when it's going slightly up. So there's no clear efficacy signal, but it shows that it's safe. You can infuse a lot of Tregs into people, and there's no adverse effects. Also, very importantly, is that also they did not change. If you look at this table here on the right, this is the infused Treg stability, because they label the Tregs with deuterium, is like heavy hydrogen. And so what you see is that all the Tregs that were injected, they stayed Tregs, because sometimes regulatory T cells in an extremely inflammatory environment, they can become conventional killer T cells. So the last thing you'd want is to infuse suppressive cells, and they become inflammatory inside the patient. But what this shows is that the important part of the table is below here. I even highlighted it, is 0% of the infused cells became non-Tregs. And so that kind of gives you hope that when you infuse these Tregs into patients, they stay Tregs. But of course, it's just safety. Does it work? Does it help with type 1 diabetes? And the answer, at least with this polyclonal Treg strategy, so a mix of specificities, is no, unfortunately, at this moment. So there's a trial, phase 2 trial now, by Collegius Biosciences, funded by CERN in California. And so if you go on clinicaltrials.gov, you see they have different metrics. I'm just picking one here, which is C-peptide. And so what you see is the patients that got placebo, a high dose of Tregs, or low dose of Tregs, their C-peptide went down regardless. And so this trial was closed. And so what they concluded is that Tregs in the polyclonal aspect, they don't really work in type 1 diabetes. People already have the disease, right? And so we can be like, well, so are Tregs really a good thing to keep working on? And the answer is, well, it is, because this trial, if you remember, it worked so well in the mice because they're antigen specific. They were laser focused Tregs. In these human trials, because antigen-specific Tregs are so rare, right? They have to be only the ones that bind to the insulin peptide, only to islet antigen, so they're very few. So people start with polyclonal Tregs. And then in the mouse, it was the same exact story. If you use polyclonal, mixed bag of specificities Tregs, they're not going to do as much as if you use antigen-specific Tregs. So the solution here really is to make human antigen-specific Tregs. So how to go about this? The insulin-specific Tregs are very rare, and Tregs, they don't grow as much as conventional T cells. So it's going to be a really big challenge to produce enough cells, right? I showed you. In people are infusing billions of Tregs into patients. How are we going to make that many cells? So one solution around this actually comes from the cancer therapy world, and that's called CAR, chimeric antigen receptor. So a normal T cell, conventional T cell Treg, it works by binding to signal 1 and signal 2 on the target cell. Signal 1 is a T cell receptor binding to the peptide MHC, major histocompatibility complex, that presents a peptide, and so T cell receptor binds to a specific peptide MHC pair. That's signal 1. Now, for a T cell to be fully activated, it needs what I mentioned before, that co-stimulation, that encouragement, signal 2. And so signal 2, again, is CD80, CD86 on an antigen-presenting cell. And so only T cells that receive both signal 1 and signal 2 become fully activated. So what was done then was to make these artificial receptors that combine everything in a single protein. So instead of having this entire protein complex with TCR, CD3 subunits, and C28, everything goes in one single protein. This CAR protein is an extracellular domain. That's where the antigen recognition happens. It's going to be an antibody-based domain that recognizes a certain surface molecule on the surface of a target cell. And then you have the signaling domain that basically mimics what the T cells usually use to get stimulated. So signal 2 will be CD28, and signal 1 will be CD3 zeta. CD3 zeta just happens to be the chain that has the most activating motifs of the TCR-C3 complex. So here's why you make sure that downstream, in a T cell or a Treg, this is a certain surface molecule, it signals, just as if it had seen signal 1 and signal 2 on a natural cell, if you will. Okay, so then with this CAR, we can now hope to circumvent the paucity, right? How scarce antigen-specific Tregs are in the natural environments. So we can kind of put this therapy in the fast lane then, right? So as I commenting, if you use polyclonal Tregs, I just showed you the data from humans, but use a schematic in the mouse as well, where if you grow polyclonal Tregs, you can have a lot of them, but only few of them actually see the antigen. In this case, organ transplant example, the transplant gets rejected. If you have a way to isolate only the antigen-specific Tregs, in this case, alloreactive, alloimmunity means other, because in the transplant setting, you can expand them enough that will be accepted. But the problem here is that Tregs don't grow as much as conventional cells, and they can become unstable, right? They can lose FOXP3 expression in prolonged growth or in certain inflammatory environments. So then this is not easy to accomplish, and this number might never be as high as we need it to be. So an alternative is what I just showed you, is take all Tregs again, don't select anything, just transduce, gene modify them all with a CAR. And then this CAR, you can design it to recognize any surface molecule you want. And then you expand those gene modified Tregs and you transfer those. And the expectation is that now those laser focused human Tregs will be able to protect the tissue that they target. So how far are we with this? This idea that basically, if in type 1 diabetes you have autoreactive diabetogenic T cells destroying the pancreas such that it can no longer make insulin, can we now bring in the CAR Tregs as tiny firefighters to stop this immune attack? So the first question, of course, will be we start in the mouse. And so the mouse, why? Because we have the NOD mouse model, non-obese diabetic mouse, number one. Number two, we've already shown in NOD mouse that antigen-specific Tregs work. So if you can replicate that success, that early success in 2004 within a modern technology field with a CAR Treg, that shows that they were in the right path. And so just to show you how, this mouse is, there's a lot of knowledge about this mouse, a lot of biochemistry even. And so this is just to show you that, I just mentioned you how, T cell receptor, cis-peptide MHC is actually more complicated than that. Not only cis-peptide MHC, it's cis-peptide bound in a certain configuration, a certain register, we call it. So the TCR of BDC 12.4.1, just a code name for the T cell receptor, it recognizes this insulin peptide bound to the class 2 in the mouse IAG7, bound specifically in register number 3. So if you have the same peptide bound in a different conformation, it doesn't work. And in this paper, for the more advanced audience, the way they demonstrated this was by forcing disulfide bonds, right? This is a way to kind of stick it in place, the structure. So now you see if you have disulfide bonds in yellow, there in A and there in B, if you see my mouse, this prove that that's the conformation that the T cell receptor of diabetogenic diabetes causing T cell sees. So we have that knowledge. So wouldn't it be great then to make a CAR, a chimeric antigen receptor, that sees this specific peptide MHC, right, as a proof of concept? Because if you can make that one, you'd assume we can now make CARs for human peptide MHC complexes that we know are important for human type 1 diabetes. And so just to show you how, vibrant this field is, there are actually 2 papers almost back to back this month of August. August 1st, the first one I'll show you, and August 10th, the second one, that actually accomplished this precisely. They took the insulin peptide bound to IgG7, register 3, R3, and they made different CARs with the antibody component binding to that. And so this is what we call a tetramer staining. That will show you these cells, each dot is a cell, and it shows you double positive, meaning that they have the CAR on the y-axis, and they bind to the insulin with IgG7 tetramer on the x-axis. So you see these cells here, these are the insulin BR3 CAR Tregs. And then the other ones are all controls to show that they don't send with the tetramer. They have the CAR, but the CAR is for something else. And so they made this, and then we have our old friends, C20 and C3 zeta, signaling domain. And so they built this CAR, and then they show, just reminiscent of the data from 2004, that this CAR Tregs can indeed prevent diabetes in NOD mice. So you see here with 2 million CAR Tregs, you can really shift the curve to the right. It's not 100% yet, but it's 50%, so it's not too far from the previous data with natural antigen-specific Tregs. So this is very hopeful data because it shows you that there is hope for us to artificially engineer Treg specificity and get results similarly potent and potentially better in the future as this gets better and better with this engineered CAR Tregs. So that was the first publication. The second one, same exact idea, independent lab. This first lab was a collaboration with the Zhang lab in Indiana and the Tang lab at UCSF. The second paper was from the Levings lab in Canada and British Columbia. And so very similar idea, independently done. So you have, again, your insulin peptide sequence and the tetramer staining proving that these antibodies bind to that IgG7 peptide complex. You can make a CAR out of that. So the intracellular domain is again C20C3 zeta. And then the binding domain is the anti-peptide MHC. And then they show that these cells proliferate specifically in response to the insulin peptide bound to IgG7, and they suppress in vitro. You see there. And then, of course, perhaps more importantly for us, they also suppress in vivo, right? So this time, not 2, but 3 million CAR Tregs, and you get a similar, 50% of the mice are now diabetes-free after being infused with these cells. So in the injection, both cases is, right before the mice overtly develop type 1 diabetes. And so this is how we are in terms of engineering CAR specific for type 1 diabetes. Now, how about humans, right? Let's go back. Okay, the mouse will show BDC 2.5 natural Tregs, if you will, work. If we have these now insulin IgG7 CAR Tregs, they work. How about in humans? So in humans, we're not as advanced, number one. Number two, the therapy in humans currently, the curative treatment is islet transplant. So you can take islets, not even the whole pancreas, you can take just the islets and infuse them through the portal vein, for example, into a patient. And if that patient does this together with immunosuppressive drugs, it now regains autonomy, like to make his or her own insulin. Now, of course, the goal would be to resolve the immunity again, right? So this now we actually have a double problem because we have autoimmune disease against the pancreas, and you have autoimmunity because this is going to be someone else's beta cells from a cadaveric donor. And so, however, this creates an opportunity to test CAR Tregs in humans, right? Can I imagine making a CAR Treg that is specific for an antigen that is only in a transplanted islets? So a good antigen to go after will be an HLA molecule, an MHC. Why? Because in humans, there are over 3,000 different MHC alleles. And so it's very unlikely that the donor recipient have the same exact ones. So you could make laser focused human CAR Tregs to the pancreas by targeting them with a CAR specific for an HLA that is in the donor, but not in the recipient. And so when we went after this HLA-A2, and then of course, for less sake of time, I'm going to present all the data, but other groups from the Levings group and Lombardi group and Jekyll group, they also made HLA-A2 CAR Tregs in these recent years. And so this is just the one I made together with Dr. Tang back at UCSF. And so what we did was we took an antibody, recognized HLA-A2, reformatted it, and it says grafted because that's a story for another hour, but it was quite complex to get this to fold properly, the protein. So we had to graft the complementarity determining regions into the right place. So once we did that, we got our anti-HLA-A2 CAR, and here's a flow plot to show that it's on the surface. The make tag there marks the surface, and activation C71 marker to show that they get activated by the right thing. So now we make Tregs that see HLA-A2. Now in this business, of course, it's as important almost what you don't bind to as what you bind, right? The last thing we need is to have a CAR Treg that is HLA-A2, but also HLA-A3, 4, 5, 6, 7, right? So we want to make sure it's specific. Luckily for us, HLA-A2 has been studied to death. So we know exactly all the immunogenic surfaces, immunogenic applets, we call them, HLA-A2 has. And so it can really screen to see which ones of those are specifically bound by our CAR. And so we did this with beads and also with islets. I showed the islet data. And so what you see here nicely with the same CD71 marker, which is a transferrin receptor that T cells upregulate, is that only with HLA-A2 in the islets do the CAR Tregs get activated. right? If you have islets that have other HLA alleles that share some of those applets, right? This 44 RME, for example, is shared with A11, A24, A31, but the CAR Tregs couldn't care less, no activation. So that's how we know that our CAR not only recognizes HLA-A2, broadly speaking, but specifically recognizes this 144 TKH applet on HLA-A2. Is based on this, a combinatorial piece of data. Okay, so we got ourselves a very specific CAR that activates Tregs. Now, how are we going to put it in if you actually go into the clinic at some point? So far, there's a lot of studies using lentivirus and retrovirus. And so lentivirus, retrovirus are great, but you have multiple copy integration and it's random. So you could imagine being a problem for safety because then you could integrate in the cancer-causing gene. You could also, not have that by having AAV, but then AAV doesn't integrate in the genome. That means that as your Tregs proliferate, it gets diluted out, right? So your final CAR Treg product would have no CAR left, and that would be a problem. So then we thought, well, let's instead do something that is not yet FDA approved, even though it might be later this year, which is Crispr Cas9. So Crispr Cas9 allows us to make a cut in a specific double strand of DNA, and then you can use that cut, the double strand break that opens up for now the cell to repair that damage with any gene you want. And so in our case, of course, we're going to do the HLA-A2 CAR gene. So here's the same exact, MIG-tag A2 CAR. It's going to be knocked into the TCR locus. And why TCR locus? Because the TCR gene has evolved to be expressed in the right amount at the right time by T cells. So if we can, hijack that regulatory gene mechanism, then that's where we want the CAR to go. So we basically reprogramming this human Treg specificity. The human Treg here used to see the world via its T cell receptor, which is, constrained to MHC class 2 bound to peptide, and now we're changing it. Now this CAR Treg can see any surface molecule I want. So in this case, HLA-A2 CAR is going to see HLA-A2 and nothing else. And so this is kind of a workflow where we get the Tregs from human blood and modify them. And then we kind of show, the same I showed before. Number one is that the knock-in works. So you want cells to be MIG-tag positive, CD3 TCR negative, and here they are, 70%. I also want to show that there's still Tregs. So FOXP3, very important. Another transcription factor I didn't mention, but is also key to identify, stable human Tregs is Helios. And so see our cells are 90% FOXP3 Helios double positive after the modification. And then, of course, they still see the right thing, right? If you incubate them with HLA-A2 expressing target cells, they get activated. Now, do they work, right? So in vitro, we do a T cell suppression assay again. And so you see this is proliferation on the y-axis. And so you see the less Tregs you add, right? That's the ratios there, 1:1, 1:2, 1:5, 1:10. The less CAR Tregs you add, the more the T effector cells proliferate. And so you see it kind of breaks down at like 1:5. That's when the CAR Treg starts struggling, right? Like these bars at 1:5 are as high as just having the T effectors alone. But so there's room for improvement always, but it does show that these A2 CAR Tregs suppress in vitro in the presence of HLA-A2. Okay, now how about in vivo? Do they work in vivo? And so for human studies, we use the humanized NSG mouse, right? These mice are immunodeficient, and so they accept human islets and human T cells, so we can study them in vivo. And so the first experiment we did was a GVHD. This is kind of a workhorse to test CAR Treg and Treg potency. And so we have 8 groups. Why? Because you deal with HLA-A2, so your blood, the white blood cells, the PBMC that induces disease, the GVHD, can be themselves HLA-A2 positive or negative. That's the 2 rows. The mice, the recipients, like our pretend patients, can be HLA-A2 negative or positive. We have HLA-A2 transgenic NSG mice. And then finally, you can either inject PBMCs alone that will cause disease, we hope for sure, or with A2 CAR Tregs, which should prevent disease. And so I have 2 graphs that kind of summarize that data. This one pulled together all the groups that have A2 CAR Tregs and ones that don't. And so you see nicely that, a bit reminiscent to the type 1 diabetes protection studies. You see roughly 50% of the mice, at least for a while, become protected from dying from graft-versus-host disease. And then we felt brave and we actually bled the mice to find the cells. And so you see here that, if you put PBMC alone, you see beautiful engraftment in peripheral blood of the mouse of CD4s, human CD4, human CD8 T cells. But of course, if you co-infuse CAR Tregs, you see very little engraftment. So the group 1-2 that is almost normal is this group here, right? So group 1-2 is one where the PBMC is HLA-2 negative, right? And so that's why. But then for these ones, you have very nice loss of engraftment. And then if you actually zoom in and get on HLA-2 negative cells, which are your CAR Tregs, look at that, not many cells, but they remain FOXP3 high. So a bit like what's shown in human is that if you look weeks later, well, in humans was a year later, but in the mice is only weeks later, you still see that the infused CAR Tregs remain Tregs, they remain FOXP3 high. So we have that. And then, of course, for type 1 diabetes, we care about islets. And so what we do, again, the same NSG mice, we actually give them streptozotocin before that will kill their endogenous beta cells. So their blood glucose spikes up. And then after that, after they have diabetes established, we pop up their kidney, and then we transplant human islets, HLA-2 expressing human islets, into the kidney capsule. It's highly vascularized site, so the islets can read blood glucose and secrete insulin in response to that. And luckily for us, mouse tissues respond to human insulin. So this rescues diabetes. And then we infuse A2 CAR Tregs to see, do they traffic to the right place? Are they deleterious or not to the islets? And so what we show is in terms of trafficking, we show that unlike polyclonal Tregs, as we kind of would expect, polyclonal Tregs don't really stay there long. They are luciferase labeled the CAR Tregs, so you can see them in the living mouse, and you see not much signal here. But if you have A2 CAR Tregs, you kind of see very nicely when you infuse them, they go first to spleen, which is on the left side of the mouse, and then they migrate to the graft, which is on the right side of the mouse. We did in the right kidney in this experiment. And so you can kind of see, if you look 2-3 weeks later, you still see that most mice have A2 CAR Tregs. And I don't show here, but these mice stay normal glycemic, meaning that A2 CAR Tregs migrate to the islets and they don't affect their function. Now, ongoing experiments we're doing are looking at whether, do the A2 CAR Tregs protect from an immune response? So the humanized mouse modeling is an ongoing by our group and others to better mimic type 1 diabetes using human cells. So we have that. Now, a different group at UPenn also did in non-human primates. So very similar procedure. You get the blood and you engineer your Tregs with CAR. In this case, they didn't do HLA-A2. They did BW6, so a different HLA allele, HLA-BW6. They grow them in vitro for quite a while, for 3 weeks, and that's the number of cells there. And then they show again, in vitro suppression and FOXP3. So you see very nicely 90% of the cells are CAR and FOXP3 positive, and they suppress PBMC in vitro. And then the idea here is, can these cells, migrate to the islets, in this case, non-human primate islets, and do they protect them or at least stay there? And so in this case, it shows the bone marrow actually as a site for engraftment, not the kidney capsule or the liver, but the bone marrow. And so what you see there is that now this is a schematic where they give the monkeys STZ, so just like in mice, to kill their endogenous beta cells, and then they infuse their islets, and then they see whether they get protected, because these monkeys are immunocompetent, right? So if you get these islets, their first reaction will be to reject them because they're not their own. But so what you see here is the Treg number spiking, and a bit like in a mouse, that population there, that will be CAR and FOXP3 double positive, 1.24%. It's only in the islets with the HLA-BW6. So that shows again that the Tregs are trafficking to the right place. And so this is data from a big animal. So now we have data from mouse and from macaque. So this, of course, informs, again, going to humans. So now in humans, there are several companies working on this. Quell, Sangamon, Sonoma, there's GentiBio, there's a whole table here from a recent review from this year. And so they all really, 2 of them are focusing more on transplant because there's just more cases, number 1. Number 2, it's also easier to start. It's very hard to recruit for trials for T1D because a lot of children, adolescents, and insulin kind of works. So it's more risky to switch to something unknown, like an engineered cell. Whereas if it's a transplant that they have to take immunosuppressive drugs that are very, they really decrease your life quality. There's more maybe incentive for patients to enroll. But so what you see is that there's 2 trials on transplant. There's one, IND, so, it's in the research phase for autoimmune disease, rheumatoid arthritis. And so I have a slide on this. I think it's actually my last kind of data slide to show from Sangamon in California, they acquired TXL. And so they're testing HLA-2 CAR Tregs. So people have been infused with HLA-2 CAR Tregs. Not for islet transplant, unfortunately, but for kidney transplant right now. And so this is kind of a paper they had where they just described the study. So there's no results yet. They're just describing you how they're going about it. What's their strategy? And so you see you have different cohorts. So it's a dose escalation like with any other drug. Again, Treg, living drug. So you have a low dose cohort, a mid dose, and a high dose. And then they're following, right? And so the idea is that they use completely replace immunosuppressive drugs like tacrolimus and sirolimus with Tregs. And that would lead, and I didn't have time to show today, but there's been some studies where they partially replace immunosuppressive drugs with Tregs, and they see an improvement in the terms of number of infections. So these patients, they have less infections, because if you can imagine, if your whole immune system gets shut down, you have really big trouble fighting infections, fighting certain types of cancer, especially blood cancers. Vaccines, of course, will not work as well on you. And so that's why there's a big push to at least partially replace immunosuppressive drugs with Tregs. And for type 1 diabetes, of course, it's the allo and the autoimmunity, right? So it's a more complex, case for this, but that's where we have it. And so with that, I'll show you kind of where we see the field heading, like what we want to accomplish in this world where we're using Tregs as living drugs to treat type 1 diabetes and other disorders, of course. And so back then in 2019, we came up with this, 4 S's, if you will. So 4 parameters that engineered Tregs really have to meet to be successful. So one of the S's is that they have to be specific. I mean, that is the whole, I think I've convinced you by now that if the Tregs are not specific, they're not going to work because there's just not enough of, active dose within that big mix of specificity. So specificity, I, of course, talked about CAR today, but, in theory, you could also isolate T-cell receptors and incorporate those instead. There's also SynNotch systems, right, where you have a dual antigen recognition. We can talk more about that maybe offline. And then, of course, I didn't touch upon that, but Tregs, they have specialties, just like conventional T cells. So there are Th1-like Tregs, Th2-like Tregs, Th17-like Tregs. And so all that means is that Th1-like Tregs seem to be better at suppressing Th1 cell responses, right? So Treg is kind of a master of disguise where he maybe partially pretends to be a Th1 cell, so he goes to the right place. Then once it's there, it suppresses the Th1 cell. And so these are engineering ways in which you can improve the specificity of Treg living drug. Another parameter is suppression. I mean, that is, again, the function of the Treg. So we need to make sure the Treg is making enough inhibitory signals, right? So this is IL-10, IL-35, TGF-beta, the sequestering IL-2, I mentioned C25. It's binding that CD86 from APC. So make sure that a Treg that this goes in has all these mechanisms intact, as is like polypharmaceutical aspect to it. We also want to survive. I didn't show data on that, but as you might imagine, any cell infused into someone's body is going to go through a lot of stress. That could be an islet cell, could be a Treg. And so how can we improve on, the survival of the infused cells? So there's some metabolism approaches, signaling pathways, such as, of course, PI3K, and then IL-2. So as I mentioned, Tregs don't make their own IL-2. So there are some ideas about combining low-dose IL-2 with Treg infusion, and they're actually engineering an orthotopic, ortho-IL-2, they call it. It's ortho-IL-2 that only binds to ortho-IL-2 receptor. So you can make your own synthetic IL-2 that only binds to your synthetic Tregs, right? So you can imagine as engineering starts, you have more and more things to play with to really fine-tune this living drug. And then finally, stability, right? As I mentioned briefly, you don't want to infuse a regulatory suppressive cell and it becomes an inflammatory cell. So how can we ensure that doesn't happen? So many groups are overexpressing FOXP3 and Helios. That's perhaps the most obvious place to start, but there's not as simple as that. For example, cytokine converters, I think it would be a neat idea, right? Where you can have a synthetic receptor where the outside sees interferon gamma, and then the inside is the IL-2 receptor signaling. So this is where the Treg, every time it sees interferon gamma or TNF alpha, it thinks it's seeing IL-2. And so imagine how that would be. A Treg that basically inflammation makes it stronger instead of weaker. And so there's all these ideas out there being worked on that make me and many others hopeful for the next few years to come. And so with that, I have my acknowledgements. A big one is Dr. Qizhi Tang. She couldn't be here today due to a personal emergency, but we discussed these slides, and I did my postdoctoral training with her and Dr. Bluestone. So it was a shame she couldn't join today. And then I also need to acknowledge my lab, of course, at the Medical University of South Carolina. They've been a fun team to work with. I've been a PI for 2 years now, and these are some collaborators we have in and outside of the university, as well as some funding from different places, including, of course, I should disclose perhaps the HIRN, the Human Islet Research Network. And yeah, I think my last slide is just, come visit us. We're in Charleston, South Carolina. We have a lot of beautiful landscapes and great science. Thank you for your attention.
Thank you very much, Dr. Ferreira. That was terrific. We have about 100 or so people here, so please do me a favor, raise your hand if you want to ask Leo a question. I see that there are a number of comments in the chat, so while we wait for people to raise their hand and perhaps come off mute, Leo, let me direct you to the first question there from Nimishi. Can you see that?
Yeah. What are the plausible mechanisms?
Yeah.
Yeah, so that's a very good question. Like, the question is, what are the plausible mechanisms by which infused Treg cells may convert into inflammatory conventional T cells? So what's been shown is that there are certain cytokines that are very good at converting Tregs into T effector cells, chiefly IL-12, especially in vitro. If you culture Tregs with IL-12, they lose FOXP3 expression, they lose suppressive function. And so the whole point of Tregs is to stop inflammation, so that you have to make sure Tregs are extra sturdy, if you will, so that they don't become effector cells and add to the problem. So the mechanism is just cytokine receptor signaling. That's a big one that we have. Another one is IL-2 deprivation. So because Tregs don't make their own IL-2, number one. Number two, they need more IL-2 than usual. So I didn't mention, but that 2004 paper was a big deal because the first time people really expanded Tregs. And it turns out that Tregs, conventional T cells, you can grow them in vitro with like 50 units per mil of IL-2. Tregs require 2,000 units of IL-2. So you can imagine how many titrations people did over the years to get to like astronomically high number of IL-2 units that you need. And so these are two ways in which Tregs can become unwanted cells. One would be inflammatory cytokines, another one would be loss of IL-2. Luckily for us, it seems from the trials from phase 1 that doesn't seem to happen. Like if you label the Tregs going in, they don't seem to be becoming, at least in the sampled peripheral blood, they don't seem to be becoming something else. Yeah.
Thank you, Leo. I'm going to switch and we're going to go to, I think, Vijay, do you have a question? You're off mute. Go ahead, please.
Yeah, thanks. I couldn't find the raise hand.
That's okay. Yeah, I think some people probably can't.
Really nice talk. Those in vivo models you show, I'm curious, do we know what the mechanism of prevention is? Is it, you showed us in the beginning, there are multiple mechanisms, inhibitory cytokines, cytotoxicity, Tregs can work in multiple fashions. What are the important ones you think in play in those T1D models? I think the answer to that will be very important for translating this and, will impact how long do we think the Tregs need to stick around to maintain effect and so on. Can you speak to that?
Yeah, because if we found one, then we'd focus on that one and optimize that one. Of course, I agree. Now, in vitro is easier because, let's say, a T cell suppression assay is really mostly IL-2. The Tregs sink IL-2, so T effectors can grow. We also have in vitro dendritic cell suppression, where you see downregulation of CD886, and there's obviously CTLA4 is what's causing that. So in vitro, it's easy to separate, but in vivo, there's so many things going on. And so in the humanized mice, it's a love and hate relationship because they allow us to use human cells in vivo, but they don't have lymph nodes. Many immune cell types don't engraft properly. And so I actually don't know. I think it's going to be a mix of, more careful studies and also just different models. I've read that a while ago, people were able to make immunodeficient mice when they overexpress TSLP. They had lymph node structures, and so you could actually, when you infuse human cells, they would have human B cell germinal centers in these immunodeficient mice. And so I think something along those lines, because lymph node is a highway of the immune system, so we need better humanized mouse models. The second thing for T1D that specifically is challenge is allo versus auto, right? All the models I showed you, it's allo, right? Is HLA-A2 as the allo antigen being recognized. I didn't look at insulin, and it's not the mouse T cells because that wouldn't make sense, right? So what people are doing to address this is to have now with stem cell, with pluripotent stem cells, it can differentiate islets, beta cells from that person, and from the same person take their T cells. Now we can actually have autoimmunity, right? If you get islet tissue, which is hard, so we make from stem cells, and T cells and Tregs from the same person. So that's what's ongoing right now in other groups, is to make stem cell derived beta cells and a type 1 diabetes person's blood cells. I think that's going to be the next round of clarity for what you're asking.
Thanks.
Leo, yep, thank you, Leo. I'm going to direct you to the next comment in the chat by Scott Carmer. If you can read that, and then if you're also having a hard time raising your hand, just go off mute, and I'll be able to see you.
Yep. Okay.
John,
so this question by Scott is. Should I go to the question, the written one first, or?
Yeah, go to the written one. I don't know who that was. Yeah, go ahead.
Yeah, so so I'll just read. So the question is, how does targeting a disease-specific epitope with a CAR Treg address epitope spreading or neoantigen-driven disease? Can this be addressed via bystander killing? So that's a very good question. So one aspect I didn't mention is that several autoimmune diseases, such as rheumatoid arthritis or multiple sclerosis, they have epitope spreading. I mean that what causes disease is one epitope, but then as the people get the disease over the years, there's multiple epitopes being targeted because there's a breakdown in tolerance in general. Now, the answer to that, so in the mouse where Tregs were first found, there's two fundamental properties of Tregs. One is bystander suppression, which means that the Treg is not going to be bound to the same cell or antigen as the T effector cell. As long as it's in the same physical location, the Treg will, it will change the APC, it will make IL-10, it will soak IL-2, and so any specificity gets, dampened in that area. The other property is infectious tolerance. What that means is that the Treg itself convinces naive T cells to themselves become induced Tregs, so that after the original Tregs we infused have died, their effect lives on. Now, I don't think infectious tolerance has ever been proved in humans, like at least with rigor, because you can imagine humans live for many decades, not for just up to 2 years. And so if infectious tolerance is true and can be induced by human CAR Tregs, that would be the answer to your question, which is even if you target just one epitope, that could be sufficient to stop the disease that is now being maintained by 10 different epitopes.
Thank you, Leo. I thought I heard the voice of Ali Naji. Am I right or wrong?
Yes. Hi, John. Thank you. Leo, great comprehensive review. I am going to echo what Scott mentioned. I'm really still confused, having to spend my life between allo with immense polymorphism and type 1 diabetes. We don't even know how many epitopes are there. This is quite a bit of, even if you look at the BDC data that you showed, which is all specific, you really couldn't get a better protection than maybe 30 or 40%. So we still are concerned about, obviously, insulin peptide is one, but as Scott mentioned, either through the spreading or even from the initial one, there's so many other ones that we don't know. And I would like you to speculate on that, because obviously the auto is embedded within the allo, which is extremely polymorphic. And I wouldn't be surprised the same applies, maybe not to the same degree with T1D.
Yeah, that's a very good question. I think the other aspects, so one is we don't know all the epitopes, and actually studies trying to see, okay, we have the NOD mouse, very defined, but if you sequence 10 mice for the Treg repertoire, you get 10 different answers. Like, sure, you're going to have some shared epitopes, such as this insulin one, but there's other epitopes that one mouse has it, that the litter mate does not. And so if you see that variation in, in inbred mice, imagine in people, because some people say the NOD mouse is just one type 1 diabetes archetype. It represents one person in the human context. And the other thing I think is also variation in just the immune response. So men versus women, different people, it reminds me of CAR T cell therapy, where if you put the same CD19 CAR T cell in different people that have CD19 expressing leukemia, many people respond, but many also don't. And there's this kind of black matter of some people that get the same cells, the same number of cells, and have the same type of cancer, but somehow they don't respond to it. And so there's going to be polymorphisms that affect, how T cells are made. And so I sometimes wonder for type 1 diabetes, if, we have these genes associated with T1D, and some of them seem to be Treg genes. So maybe T1D Tregs are intrinsically defective. It's been hard to prove this, right? Because once you put T1D Tregs in vitro, they actually work okay. But yeah, you put them in so much IL-2, beautiful medium incubator control. So just because they can work in vitro does not mean they're not defective in vivo in the patient. And so sometimes I wonder if there'll be a room for allotherapy, or we take Tregs from non-diabetic patients and maybe make them universal if you have to, right? The hypoimmunogenic, that's a new wave now that I didn't mention, but that could be one way. You have Tregs from people who don't have T1D. So we know for sure that Tregs are not defective in any way, you think, at least in terms of T1D, and then infuse those Tregs into the T1D patient instead of their own. And maybe they'll be more powerful, because we just don't know where the problem is in T1D. So multifactorial, something about the immune system in T1D patients leads to that disease.
Yeah, thank you, Leo. Unfortunately, Shiva and Sigmund, I think that's going to be our last word, but we'll see if we can get the questions to Dr. Ferreira for him to answer to you at a later time. Thank you, everyone, for attending this meeting. It was a terrific webinar. Leo, again, thank you very much for your time, and we'll make sure to get these questions to you.
Yeah, thank you. These are good questions. Yeah, thank you.
Yeah, we'll do that. Okay.
Thank you.
Bye-bye, everyone.
Thanks.
Thank you.